Sorry. Can anyone hear me? Hang on, I'll just hold this a bit closer up. That'll be fine. So yeah, I am Will Dawson. I am a second year PhD student, literally about 20 metres away up the road. Some people in the audience may know me as the soil boy. So today we're going to be talking about soil, and without further ado, you may question, why soil? So when you think of the most biodiverse habitats on the planet, your mind may go towards the rainforest, like the Amazon, or Great Barrier Reef, but in fact, 59% of all species on the planet are found within soil. It's not just this dark, inert, silent little place, it's packed with all forms of life. At the base, you've got the bacteria. If you did want to tick off on your bucket list that you've discovered a new species, 85% of all bacterial species within soil are currently undescribed. So if you wanted to cross that off, get into that. Then you've got the poster children, the fungi. I will not be, unfortunately, talking about fungi today, and that's because they deserve their own absolute evening. Just know that they're really, really cool, but I work on animals, so we're going to talk about them, I'm afraid. Right at the bottom, you've got the microfauna. So if you take every animal on the planet and averaged out their leg number, you may think, okay, it's going to be like 5.4 or so because of the amount of insects everywhere. Actually, the average number of legs on the planet are less than 0.1. And that's mostly because of these guys, nematodes. The tiny little ones, some of them are really bad for us in diseases, but most of them are within soil. But most of my research isn't on them because they're an absolute horror show to identify. So, then we get into my kind of stuff, the mesofauna. These are your mites, your springtails, and anything nice. So springtails have 6 legs, they look like an insect, they're not an insect, they've only got 2 body parts rather than 3. As the name suggests, some of them has this like lever thing on the underside of the abdomen, which are used to catapult itself away. If a springtail was sized up to us, it would be the equivalent doing a standing backflip over the Eiffel Tower. They're really, really awesome. As well as that, you've got mites, they're everywhere, they're absolutely fascinating. This is one of the box mites. They look like a little armoured tank. They are everywhere. Guarantee you, they're walking around the uni right now. Then you've got the macrofauna. These are your worms, these are your other insects. And finally, you've got the megalofauna. The annoying things with 4 legs and fur that I don't work on because everyone thinks they're really charismatic, which they are, but insects are what makes the world go round. So unfortunately, we're not going to be talking about moles. So, I've told you the players, and now we want to look at the services they provide. So, one of the main things they provide for us is climate regulation. The UK soil holds as much carbon as the entire human population releases as greenhouse gases every single year. If we want to help mitigate against climate change, we need soil biodiversity on our side. As well as this, it's important for all terrestrial life, not only because the majority of life is found within soils, but almost every single terrestrial habitat on the planet has soil in it. If we don't look at soil, you're kind of neglecting the whole foundation of all of these habitats, and yet they're tended to be forgotten. It's also really important for the water cycle. We hold as much water within our UK soils as all the UK's rivers, lakes, streams, ponds, reservoirs, all combined. Finally, it's really important for us. We're a very hungry species, and 98.5% of our calories globally come from soil. In a world as tumultuous as it is, if we had tumultuous soil, I don't think it would be actually any better for us right now. So we really need a healthy soil system in order for us to kind of, at the moment, get by. So we look at these services and we want to think, what can everything do to kind of provide it? And you're like, I want to look at carbon sequestration. Hopefully fungi can do that. I want to look at agriculture. Hopefully I can just look at the macrofauna. Unfortunately, everything is all interactive. Everything is important for everything. But for the sake of this talk, because otherwise I will be here until Christmas, we're just going to look at the insects today and other invertebrates. So, you want to look at invertebrates, how do you go around doing it? Basically dig up a bunch of soil and then you have to extract everything out of them. The classic way we do that is with the bottom apparatus here, called a Talbot funnel. it is a mesh with a cone going down. And most soil invertebrates are kind of a bit gothic. They hate light, they hate warmth. So you turn a bulb on above this soil and everything tiny crawls away down that mesh into your cone and your collection tube, which is really great when most of your things are less than 1 millimetre in size. So you've got your community, you now want to go around IDing it, and that's where all of the fun begins. I say fun because it's lots and lots of hard work. Taxonomists on the whole is a dying kind of field. It's got loads of demographic problems, very short in expertise in a lot of areas, but particularly soil-borne. So you need to try and identify everything, and most things you can't identify. If you're taking a fly species where you have to look at the number of hairs or one part of its wing, then you look at its baby, its larvae, that's literally just there to eat it, eat it, and eat. That doesn't have that feature anymore. You're a bit stumped. So we need to think about more accessible approaches, cheaper, more time-efficient approaches to monitoring soil biodiversity. And that's when we can look at ecoacoustics. I can already see a few faces like that, which is great news. So, ecoacoustics is the study of an ecosystem's soundscape. This is classically done in birds, bats, increasingly in aquatic environments too. Some really interesting aquatic ecoacoustics work. If you wanted to increase the speed of a coral reef forming, you can play the sounds of a busy coral reef next to a fledgling one, and it will attract everything in. Not just fish, what you think have ears, but also just coral, which you think, how the hell does that hear it? Who knows? I think they're trying to find out. But for my talk, we're going to be talking about soil, because in the last 5 years, it's been an absolutely expanding field. Unfortunately, the sound of soil is not like these lovely ladies. In fact, it's more along these kind of lines. So the classic setup is a contact microphone, which you stab a little probe onto it, put that in the soil, and it transduces up all of these sounds. Most insect sounds only travel around 30 to 50 centimetres within soil. Any further than that, you're probably more looking at weather-related sounds or human-derived sounds. So you've got your sonograms, just like you would have for any kind of sound recording, and you can start to kind of try and process that data. Over on the one on the left, you can see in the box, there's a bunch of activity going around at once. It wasn't just because there was a soil invertebrate's birthday party where they all went around and had fun. That was just a plane flying overhead. So by able to comparing mic to mic, you can start to realise what's caused by biological sounds, what may not be. So again, on the right, you can see at the start, some of them kind of coordinated. These microphones were 4 meters apart. So if that was made by a worm, that's one extraordinarily loud worm. So you then look at what things are different, and you can start to look at the insects and invertebrates that way. So as I said, ecoacoustics used in a variety of settings, especially in above ground, but increasingly in soil. One of the loudest things you'll come across are earthworms, which are probably only beaten by the moles trying to eat the earthworm that's going next to your microphone. Earthworms have this really characteristic kind of like gurgling sound, which is really useful for tracking them. In the UK, we like to think of earthworms as a farmer's best friend, which they are. They're absolutely great. However, earthworms are also an extraordinarily damaging invasive species in some parts of the world, such as tundra habitats. We love worms in our farms because it aerates the soil. It turns it over. However, if you've got a tundra habitat, you don't want that to be happening. That transforms it from tundra into grassland. So we can use acoustics in order to track these progressions, these populations of earthworms, to try and manage these invasions. Also, you can use it as a diagnostic tool for pests. Some things are very loud when they chew on your roots, not your hair, don't worry, I mean your plants. So if you had a vineyard and you wanted to track a weevil feeding on it, you can start to try and listen to it. And increasingly, it's being used as a proxy for biodiversity. A lot of research has been done within spring, summer months, and we'll take a few meadows and you'll be like, this sounds way more complex, this has way more sound going on, and it correlates to more things living in it. So, pro, there's a correlation between biodiversity and ecoacoustics. Great. Unfortunately, it's not that simple. Soil is a really weird place. In winter months, for example, your biodiversity may actually increase. And that's because if you're an insect and you wanted to overwinter, you want to pupate, soil's a very good real estate to do that in. So you may actually go around monitoring your soil about winter, and you find that your biodiversity goes up. However, they're doing soddle, which means you hear soddle. So therefore, your acoustic complexity actually goes down. So in that case, what you're looking at is the activity of everything. Once you start to look at the activity of everything, you can start to look at their interactions and how they interact with one another. So, interactions and networks are extraordinarily important. Let's say everyone here is a different soil invertebrate, and there's some giant entomologist looking over us and go like, great, we're really biodiverse down there, fabulous. But if our food web or our interactions are only really channeling, let's just say, through this kind of back row here, and suddenly a drought comes along, or a new fertilizer, and that back row can't really be asked to be here anymore, they may just bugger off, and that means the rest of us are a bit scuppered. What we want are really well interconnecting, really robust networks. If it's robust, it can withstand climate change or ecological change way, way easier. So we look at food webs within soil as well, a classic one of that. Gone are the easy days of a grass going to an arrow to a rabbit, going to an arrow to a fox. In soil, they're absolute bedlam, but they're really important to look at for those reasons I just stated. You can also look at ecosystem functions, as well as the services of those food webs as well. So you want to look at food webs, how do you go about doing it? You could go down the stable isotope route. You can kind of like monitor how the carbon and nitrogen transfers from every individual organism, or you can look at something called dietary DNA metabarcoding, which is a lot of jargon. However, it's basically sequencing what's in their stomachs, what they've been eating. But like I said, you don't want to do that traditional method because it's really time consuming, and yet you have to. You have to extract those invertebrates out, you have to ID them, and then you have to mash them up or dissect out their stomach contents and sequence that DNA. And that gets very expensive very, very quickly. So that's where my PhD comes in. So I want to see, can we infer these networks using that really cool acoustic data alongside other genetic methods. These genetic methods are called environmental DNA. Organisms on a whole, we're really messy and careless with our DNA. We shed it absolutely everywhere. So if you took a bunch of soil, you can sequence whatever DNA is left within that soil to kind of get a shopping list of what's been there. It's this thing, it's this thing, it's not that thing. That's really useful, but what it doesn't tell you is what they're doing. And that's where hopefully ecoacoustics can come in. It tells you the activity, it can tell you what they're doing. So, I've got a few kind of plans in that, thankfully. As all PhD students know, I've got the bane of my life, a literature review, out of the way. So, I'm concentrating at the moment on my experiments. The first of which is seeing how do food webs vary within soil. What causes them to change? How do they change over time? And can we use these ecoacoustic methods within soil? And the final one is over in rewilding. So, just north of Morpeth, Cockle Park Farm exists. It's tied to the university. It's a really, really great research place. And if you go in the summer months, you'll maybe see one field that's particularly odd. It's got 14 different belts of different sized grasses, different colour, different densities. And that's because every single year there's been 14 different manure or fertilizers applied. That's been going for 130 years, thereabout. It's one of the world's longest running experiments, and it's literally just up there in Hebron, just north of Morpeth. So, with ecoacoustics, I've already said there's a slight limitation that they're often just done in spring and summer. So I'm going there in the depths of winter, all year round, to kind of track the changes across the year. So I'm seeing how fertilizers impact soil invertebrates, how it impacts their food webs, and whether we can use ecoacoustics to show it. Finally, we've got Castle Howard, just down by York. Actually going there tomorrow. Hopefully the weather stays like it is today, but I'm not very optimistic. It's a big private estate. They've got a bunch of arable land that's very wet, very boggy, very difficult to grow things on, and they're giving up to rewilding. This amount of land they're giving up is as big as York's inner city walls, for context, and they're trying to triple their biodiversity in the next couple of decades. Also, it is a place where they filmed some of Bridgerton. I haven't watched it, my mum absolutely adores it. Unfortunately, they haven't tried to get me in because I'm not being caked in mud whenever I'm down there with a bunch of buckets, but I'm holding out hope. So, rewilding is such an amazing buzzword in ecology and conservation. It's a really vital tool. But one of the biggest problems with a lot of rewilding is baseline data. It's all good going to an amazing place like NEP and looking at the soil biodiversity and being like, wow, there's so much, but we don't know where that's come from. It could have been wow all along. So getting this baseline data at the start of rewilding means anyone in the future can look at it and be like, this is the impact it's had. And yet that's never actually been done in soil. So every 3 months I go down to lovely Castle Howard and monitor the soil communities, their acoustics, as well as their food webs. So I've got a shrubland plot, grassland, woodland, and an arable control, so hopefully we can start to track the impacts of rewilding upon soil biodiversity, which is simply not being given enough attention as it has. I think I'm running out of time, so just wanted to make a few remarks. Hopefully you can take away that soil is not just this really disgusting thing, it is absolutely awesome. But food webs is vital for everything. It's all good to just look at the basic kind of, this thing's there, but how do they interact? How robust are all of those interactions against stuff like climate change or habitat destruction? Also, the ecoacoustics are pretty cool, especially in soil. There's a lot of dirty talk going on in soil, I can tell you that. But hopefully, watch this space and I can actually give you something back in the future. I've got to shoot off in a bit after this talk, so I may not be able to see everyone afterwards, but I'm happy to answer any questions now. Thank you for being a lovely audience. Ask away.